Hello, and good morning from Peru. Thank you, Desi and Sophie, for the invitation to share our work and to introduce ourselves. So I realize this journal club is very broad in its background, so feel free to ask any questions at any moment. And it will be a very short introduction to what we do and one particular project, which is very close to my heart because it's the very first project that got started in the lab as the lab got started. So I'll tell you a lot more about it. So one focus of my lab since coming back to Peru in 2017. First, I should say I trained here in Peru in Cayetano, Heredia. I then did my training in molecular microbiology in the United States, Washington University in St. Louis, and then did some training in public health to return to Peru about 3 years ago to start a position here at my lab. And then Guillermo and Alejandra are also some of my first students. Guillermo is actually the first person that joined my lab, and Alejandra joined about a year later, and they have both been involved in this project. So this is the lab as of a year ago. Now people are working from home, but it has grown substantially, and now we're covering a range of different organisms and projects. But we started from from scratch, and this is something we sent out a couple of papers. One is what came out of my graduate thesis in 2016, and this comment on Nature Microbiology earlier this year through my colleague Maya Nadimpali from Boston University. No, sorry, she's at Tufts in Boston. Sorry. And this comment and this point that we've been trying to communicate for a while, that informal urban settlements, or people call them slums or shanty towns, how this can be a hotspot of antimicrobial resistance. And this is important for countries like Peru, where a large portion of the population lives in such conditions. So we're arguing that these settlements are at increased risk for environmental AMR transmission, not just from medical use, and that it's worth looking at these webs of transmission and an exchange of antimicrobial resistance genes. But to give you an idea of where we start from this premise that AMR is not just a clinical issue, of course, and because of various biological interactions between different agents, it's more of an ecological problem of AMR resistant vaccines moving around between different environments, directed by different selective pressures. And so, of course, we think a lot about antimicrobial usage that goes into humans and animals, but also they exchange these AMR genes through where this human feces go, animal feces go, and all where they will end up eventually. Different environments are exposed to these, different water bodies, and also through different animal products. We'll come back to this slide several times throughout the presentation. Also, in different environments, and depending on how abundant and the opportunities there are for exchange, different resistance genes, given the pressures that are present, are prone to moving around between different environments through horizontal gene transfer. And this is very basic illustration showing that depending on how abundant they are, and also chance, what originates in one environment ends up in a very different environment over time. So this is where we are. This is where we live, Lima, Peru. We are right next to the ocean. So if we look at Lima, this is what it looks like from one of our top of our highest vantage points. And it's very urbanized, and it's a very old city, around 500 years of history. But if you start going to the outskirts of the city, a city of 10 million, this is what you start seeing. We are located in a very long strip of desert right next to the ocean. And as people have moved into the capital in the last 3-4 decades, they start establishing these settlements. Right? And over time, they grow. And this is what I was trying to. Can you see my pointer? Yeah. Okay.
Yeah.
Cool. So most of the population is clustered around here. Our lab, our university, is in the north part of town, and our study site in the south part of town. And when I told you about the outskirts of the city, I mean this area around here. And it is estimated that something between 3 and 4 million people live basically in these conditions, right? Rolling hills all around the city, built with limited access to basic infrastructure and services. So it is relevant for our reality. And as you go up the hills, you start seeing newer settlements and less access to basic services, to water, electricity, sanitation. Okay, and this is of course not unique to Peru. This is a situation that's representative of many places in the world where you get this combination of poverty and overcrowding. And based on the comment that we shared, we have different sources of AMR in these communities, and also different behaviors, different routes through which AMR can jump into humans that are particularly relevant to the slum environment because, again, overcrowding, limited access to water, contact with animals. And we'll just highlight a couple of these. And this is showing you photos of our site, which we've been conducting studies at this site for the last 30 years or so through our collaborator, Dr. Bob Gilman from Johns Hopkins. Right? So of course, overcrowding and poverty, that's what comes with this. If you put a lot of people sharing the same facilities, latrines, then you get the idea of how much fecal oral transmission is happening here, especially in babies. And of course, being a desert and not having many houses not having drinking water, a lot of them buy water from vendors, from trucks, and they transport them to their houses, and there's increased risks of contamination. A lot of water gets reused, and you get the idea, right? When there's limited water, then there's to be less hand washing, and that's always an issue in these settings. Of course, animals are an issue, domestic and what people eat. There's lots of stray animals coming in and out of houses with their own microbes coming in and out. There's feces sometimes all over the place. And then some people grow their own animals for consumption, and a lot of people also buy them in local markets under, let's say, unsanitary conditions or less than ideal conditions. So you start to get the idea of how these transmission networks are happening, right? And that was the main goal of my project when I was a PhD student in the lab of Gautam Dantas at Washington University. So we were trying to put numbers on this and characterize different environments and use metagenomics to identify resistant genes in different environments and try to start getting a picture of how connected these are. And this is the paper that came out in 2016. This is honestly the paper that gave me the job at Cayetano and coming back and opened a lot of opportunities. And this would be a nice summary in that we see the presence of very well-known resistant genes in very different genetic contexts, which is a strong proof that there's a lot of horizontal gene transfer going on. And this hairball is basically the collections of resistant genes or resistomes from people, from sewage, from different environments, from domestic animals. And the message is basically that everything is everywhere. Right? You see the same genes present at 100% identity in humans, in domestic animals, in very young babies, in the sewage system. Everything is everywhere. And it's, we believe, facilitated by all these risk factors that I just explained to you. Okay, and one of the interesting observations. We did some functional metagenomic screening, and that is to identify unknown antimicrobial resistance genes that might be present in the gut microbiota. Right? Some of these bugs are not culturable, so we're trying to look at unknown resistant genes that are present there. And we found, of course, resistance to beta-lactams, which are very commonly used, to tetracyclines, which are very commonly used. But chloramphenicol, over and over again, it showed up as the chloramphenicol resistance phenotype and genotypes kept coming over and over again in human guts. And this was puzzling because chloramphenicol is not used anymore. It hasn't been used in the clinic for more than 20 years. And the fact that we're seeing high rates of resistance to chloramphenicol, even in babies, 1 year old, it was an interesting observation. And we're wondering where it would come from. Because, well, it's not been used. So maybe the pressure is still around from somewhere else. Maybe it's from the environment. So it's a very simple question that we posed. And is food related? Right, and this was because we had already looked at the other environments, and also a very practical reason when we started the lab, and a couple of just very broad questions. And is how much resistance can we find in these sort of chickens in this environment, knowing that chicken farming in Peru uses a lot of antibiotics? It's very poorly regulated, so lots of antibiotics go into chickens. And if these chickens are a potential source of antimicrobial resistance genes for humans, right? So that was the first spin-off project in my lab. And this is a study that we are wrapping up now, and we are getting ready to find it a home in the next few weeks. So again, this is the first project that came out of the lab. And we started very basic, right? When one starts a lab, we didn't have any equipment at all. And I knew I wanted to do something related to genomic epidemiology, or using whole genome sequencing to track different environments. And we had to start somewhere, and we have to start growing bacteria because we just got our incubators. So the easiest thing to grow was E.coli to get things rolling as we would purchase our sequencer. And first easy project, low-hanging fruit, was isolate E.coli from everywhere and see what differences you find. So conceptually, it's very simple, this study. And that's what we tried to do. And again, this is Guillermo, grad student in the lab doing his PhD, and Alejandra, veterinarian working also in my lab. And this is the team Pollos, as we call it. Why chickens? Because Peruvians, people in Lima especially, we eat a lot of chicken. Right? I think Brazilians eat slightly more chicken than we do, but it's still about 40 kilos of chicken per year per person, and it's even higher in Lima. So it's a lot. And if we assume that a lot of the chickens are raised under these standard farming practices that use lots of antibiotics, then you can sort of see where there's a potential risk coming in through the food chain. Lima is the main poultry producing region, so farms are not far from our study site, and all these chickens get distributed all over the city. Okay, so very simple design, which just went into different sources and that are related to chicken exposure as we first set it out. So we went for adults in the community and their babies, and this is through a parallel cohort study that was running. So we could get access to adult fecal samples and baby fecal samples, assuming that babies would get a lower environmental exposure to AMR. And then adults that handle these chickens in the markets, we convinced a few of them to give us some samples, and then we compared the market chickens And also we found access to one farm far outside from Lima that raises chickens without antibiotics. We wish we had more of these farms, more of these isolates, but we could only get access to one. So you can see we got chickens and humans and sort of a gradient of chicken exposure, and that was our basic design. And this is, yes, plainly isolated E. coli, and we did some disc diffusion assays to see how resistant they are, phenotypically resistant to different drugs. These are they call them free hens, so they grow freely without antibiotics. So we sampled those, and we got our collection, and this is what we got. And these are our first set of results. This is just rates of antibiotic resistance by group. And what already stands out is that market chickens are pretty much resistant to everything by a wide margin. So the common ones, tetracycline, amoxicillin, sulfamethoxazole, chloramphenicol, and florfenicol, which are, chloramphenicol was very commonly used in chicken farming until very recently, when they got banned, sometimes 6 or 7 years ago. They got banned, and then florfenicol was introduced. But chloramphenicol has been used for a long time, and so that's the first hint of where we thought resistance to chloramphenicol in humans might be coming from. And well, you get the idea. Highly resistant to everything. A couple categories in which the chicken vendors tended to have higher levels of resistance happen in florfenicol, which is some weak evidence that there's something going on with florfenicals and chickens and how it's spilling into humans. But we'll get into that later. And if we look at multiple drug resistances from the highest numbers to zero, you'll see again that chickens, conventionally raised chickens, are multi-resistant to everything you throw at them. And this is pretty scary to look at. And with the organic chickens being the most sensitive. So there is already some evidence, even though it's the sampling is limited, that something, of course, in how they were raised definitely affects the E. coli that are present in them. From those, so we had to start sequencing, because that was one of the goals from the lab. And we selected a set of 118 E. coli isolates from these 315. And we tried to get equal representation from all 5 sampling groups, and a collection of MDR plus sensitive isolates, so we can put them all together, compare them, and see what genes are present and how they relate to each other. So we had selected 118, got a nice mix, and first we classify them by the sequence type that they belong to. So this is broad distribution of sequence types in chickens and humans from all 5 groups. And the first thing that we can see is that these ST10 and 155, 48, and a couple others sequence types that are shared between human and chicken hosts, right? Which already suggests that these are fairly promiscuous between different hosts and can equally survive in chickens or humans. Especially ST10 is worth looking at. And then we got some minor groups, sequence types, some of which are common, some of which are not. And we could see that some are clearly clustering exclusively in chickens in both groups, and some are clearly clustering in humans. And also different humans have different sequence types of E. coli. And this is not surprising, of course, E. coli being so highly diverse and present very much everywhere. We did see very high rates of resistance, as was already hinted by the phenotypes. And so if we look at individual resistance genes by matching our genomes to what was available in CARD, you may be aware of the resistance database. Of course, we see a lot of hits in the market chickens especially, right? And some in the organically raised chickens, but of course a lot less. And then humans have less than the chickens. But we already saw the phenotypes, which recapitulating in the genomes of these isolates. Okay, this is just a very long list. I don't know if you can even see the legends, but we see a very large diversity of genes, including what the ones conferring resistance to chloramphenicol, florfenicol, to all sorts of beta-lactams, and you get the idea. We saw a couple hits to KPC3, carbapenemases of clinical relevance, so it's scary that you can find that in a chicken, but not surprising. We have seen this before in other studies. Also, the colistin-resistant gene MCR1, which has already showed up in chicken isolates all over the world. And well, you get the idea. If we put all these in a phylogenetic tree, just to show how they relate to each other, it's sort of the same message. That's there's some clustering of chicken E.coli, there's some human E.coli, and there is some mixing here, right? This ST155, you see that there's chicken and human isolates that are somehow related. You see something similar in ST10. So again, they're mostly chicken or human STs, but you get some jumps, right? And they are able to, we believe, stably colonize human guts and chicken guts. But if we count SNPs here, it's not that they're the exact same E. coli. They belong to the same ST, but they are fairly far apart in terms of their core genomes. But they can still be classified as the same sequence type. Okay, and if we look at individual genes, and I told you about florfenicol resistance because it's so interesting that humans rarely get exposed to florfenicol. Well, they're not supposed to be exposed to florfenicol directly, but we see these florfenicol resistance genes jumping in and out. And we see the same gene, 100% ID, surrounded by different mobility-related genes, which, I mean, I wish we had long read data to map, to better map plasmids. And this is short read data, and you know that comes with some limitation, but at least we're seeing that the same gene is present in very different genetic contexts. And again, this is some evidence that there's something about florfenicol that it's maintained in these populations. It makes sense to have it in chickens because they're so exposed to it, but where is it coming from in humans? And it's very hard to make that connection, but at least we have some evidence that it could be due to some sort of spillover from chickens. And sorry, I just pasted a lot of text here, but the summary again. Lots of E. coli, lots of resistance in chickens, a lot more when they were exposed to antibiotics. We see very high rates of resistance in babies as well, and which sort of tells you that, again, it's not that babies are exposed directly to a lot of antibiotics, but they are probably exposed to a lot of antibiotic-resistant bacteria from the environments, and most likely from their family members. Well, lots of resistant genes across the genomes, and again, this issue of florfenicol and where is it coming from, and is it a marker of resistance that we can find in humans, but we can trace back to chickens? And that's sort of the question that we're trying to hint at, but it's hard to prove. Again, and that's again brief study. This is our lab once again. There was a field team again through our collaborator. There's all these nurses and workers being part of the clinical studies being carried out here. And of course, the families that were very kindly opened their doors and were open to the studies. So yeah, that's it. First paper from our lab coming out soon. And again, it's hard to prove a lot of these connections, but we're hoping that we're providing some evidence and shining some light into how worryingly it is that chickens everywhere that people eat have such high levels of resistant genes. So that's where I stop.